Okay, fabulous. Yeah, so today I'll be talking about the role of climate change, dust generating events, and rodents on the emergence of valley fever in California. Um, and as just a routine disclaimer, a lot of the work that I'm presenting here was done in collaboration with California Department of Public Health and EPA, but the results do not necessarily represent their views. So I'd like to start with my acknowledgement slide, just so that everyone can see that the work I'm presenting is really the product of a huge body of researchers. I've been at Michigan for about 2.5 years, but was previously at Berkeley, as was mentioned, and so a lot of the folks listed here are from Berkeley, as well as from California Department of Public Health, who we collaborate closely with. And then much of the work that I'm presenting is funded by the NIH, including an R01 award that is PI'd by Justin Rameis and John Taylor at UC Berkeley, as well as an FNK award. So my research interests are really motivated by the fact that we know that human pathogens can be aggravated by climate change. And we don't often think about fungal pathogens as climate-sensitive pathogens, but they are increasing in importance as a global problem. Recently, the World Health Organization released a list of priority fungal pathogens. There are a lot of concerns about the emergence of new fungal pathogens under climate change. And in the 5th National Climate Assessment, various fungal pathogens, including valley fever, were highlighted as key examples of climate-sensitive infectious diseases that we should be concerned about. So the specific fungal pathogen that I'm going to be talking about today is known as Coccidioides, which is a pretty hard word to say. It is often abbreviated cocci. Coccidioides causes a disease known as coccidioidomycosis, which is known colloquially as valley fever. So throughout this presentation, I might use these terms interchangeably, but it's all referring to the same disease. The disease is endemic to the southwestern US, particularly California and Arizona, but also parts of New Mexico and Texas, and does seem to be expanding across the western US. And it is a fungus that grows in the soil, typically arid soils, and then the spores can become aerosolized and inhaled by a mammalian host, where it enters the lungs and can replicate, causing respiratory disease. So coccidioidomycosis is considered an emerging fungal infection because we have seen dramatic increases in the infection over the past several decades. If we look at just California, a lot of the incidence is concentrated in sort of the lower part of the southern San Joaquin Valley, which is shown in this darkest red county here, that's Kern County. And if we look at incidence over time, over the past 2 decades, we can see that in that highly endemic county, incidence has increased about 8-fold. But perhaps more concerning, if we look at some of these coastal counties and more northern counties where historically the disease has not been as endemic, we're seeing about 15-fold increases in incidence. And when we look at Arizona, the picture is quite similar, that the highest rates of increases are outside that region that we have historically considered to be the most endemic. And there is a lot of concern that climate change is playing a key role in this. And if we think about the life cycle of the fungus in the soil, we can start to conceptualize how climate might play a role. So in the soil, these fungi grow as long branching chains called mycelia. And as with all fungi, they grow when they have access to nutrients. And we'll talk about what some of those nutrients are. But nutrients and moisture help the fungus grow. Every other cell develops into a spore. And then when the soil becomes dry, those mycelia become brittle, which allows those mycelia to break and release individual spores, which could then be carried airborne and inhaled by a host. And so because of this process, we are concerned that airborne spores may be carried in fugitive dust emissions, and that people who work outdoors and dig in the dirt might be especially susceptible to infection. We do see several disparities in who becomes infected with valley fever as well as who dies from valley fever. So we see higher incidence and mortality among people who do work outdoors or with soil, including construction workers, agricultural workers, firefighters. In California, there's high incidence rates among people who are incarcerated, potentially related to where prisons are cited in the state. And then we do see higher rates of infection and mortality among African American and Hispanic individuals. So today I'll talk about three separate areas of this disease through an overall research agenda is trying to understand the environmental factors that affect the emergence of coccidioidomycosis or valley fever in California. So I'm going to start by talking about drought and other hydroclimate swings that the state has been experiencing in recent years. So the first research question that I will ask is what is the causal role of precipitation, temperature, and drought on emergence of valley fever in California? And this is work that is not super recent, but I think it's helpful just to kind of set the ground in terms of understanding how some of these basic meteorological precursors are affecting incidence. So first, we looked at how prior sequences of temperature and precipitation affect regional trends and incidence within the state. So we partnered with California Department of Public Health to obtain information on cases of valley fever that were reported in California since 2000. It is a reportable disease in the state, so any case that gets detected should be reported to California Department of Public Health. We aggregated these cases to the census tract level, and then we linked the census tract to data on lagged environmental conditions, including temperature, precipitation, other environmental covariates like soil texture, and then spatiotemporal variables. We then used a distributed lag nonlinear modeling approach to understand the relationship between lag temperature and precipitation and incidence. And so this is a nice approach because it lets us look at both nonlinear and delayed effects of temperature and precipitation on our outcome of interest, which is valley fever. So for each of the lagged temperature and precipitation variables leading up to the time of disease onset, we were able to look at this nonlinear exposure response relationship. And we built a model for each county and then pooled the exposure response relationships across each county. So the graph on the left here is showing a single exposure response relationship at a given lag, and then the graph on the right is showing the effect of a 1 unit interquartile range increase in precipitation on the incidence rate of valley fever in California. And so if we look at the graph on the right, we can see looking at a lag of 3 months, if temperature is increasing from the 25th percentile, which is 0 millimeters, to the 75th percentile, which is about 2 millimeters, we see a decrease of about 5% in the incidence rate of valley fever in California. So that IRR of 0.95 is getting plotted over here on the graph on the right for the lag of 3 months. So just to kind of orient you to these plot dimensions. So here we're showing a plot on the x-axis is the lag, so months prior to estimated disease onset. The y-axis again is the incidence rate ratio associated with a 1 interquartile range increase in precipitation. And then we're specifically looking at disease onset in the fall, both because that's when we see the most cases of valley fever in California and because that allows us to map these lags onto specific times of years. So what we found was that in the short term, so in the roughly 3 months prior to case onset, more rain is associated with fewer cases. And this makes sense because in that short term, that's when we expect the soil to be drying out, the spores to becoming aerosolized and emitted in fugitive dust emissions and inhaled by a human host. However, in the kind of 6 to 9 months prior to case onset, we see that more rain is associated with increased transmission. And again, this makes sense because this is the period of time where the fungus is growing in the soil and requiring access to moisture and nutrients to grow. And so this lag corresponds roughly with spring and winter prior to onset. When we look at longer lags, we don't see consistent relationships as main effects, but what we do see is that precipitation in the 1 to 3 year prior to case onset is an important effect modifier of the spring and winter precipitation. And specifically, if we see dry conditions in the 2 to 3 years before case onset, the effect of that winter rain becomes more pronounced. So winter rain following a dry period amplifies valley fever incidence more than just winter rain following a previously wet period, and we can talk about why that might be the case. So once we kind of established that there is this sort of sequence of rainfall conditions needed whereby wet winters followed by dry summers are associated with incidence, we wanted to then understand the effect of drought on incidence in California, knowing that California is plagued by persistent drought conditions. So for this, we used an ensemble modeling approach where we came up with a variety of candidate algorithms to predict the number of cases of valley fever per census tract as a function of temperature, precipitation, other environmental conditions. These candidate algorithms included some pretty simple generalized linear models, some nonlinear generalized additive models, as well as random forest models. We then used a leave-out one-year cross-validation approach to determine the out-of-sample prediction error for each of these candidate algorithms, and used that to calculate a weight for each of our candidate algorithms based on how well they were performing. And then we computed a weighted average of all these predictions so as to minimize our out-of-sample prediction error. And so what you're seeing on this graph here on the x-axis is the year. The dots are representing the monthly case count in the specific county of interest, since we built these models for each county in California. The line is our ensemble model fit to the data. And then the grayed out periods here are 2 periods of extreme drought in California, including 2007 to 2009 and 2012 to 2015. So on this graph alone, what you're seeing is how well our ensemble model is predicting the data. We then use this G-computation approach to ask how many cases of valley fever would we have expected if those 2 droughts in California had not occurred. So essentially we set temperature above average to the average and rainfall below average to the average, and then plugged those conditions into our ensemble model to generate these counterfactual predictions of how many cases we think we would have observed without the drought. Because we saw that antecedent rainfall could have effects on valley fever up to 3 years prior, we looked at both predicting cases during the drought period as well as in the years that followed the drought period. And so what you're seeing here is our model predictions in the colored lines for what we think would have happened in California if the drought had not occurred. So in green is if the 2007 to 2009 drought had not occurred, and in pink is if that second 2012 to 2015 drought had not occurred. And what you can notice is that during the periods of drought, we actually expected that more cases would have observed if the drought had not occurred. So we see a suppression of cases associated with the drought. But if you look at the 2 years following drought, we see kind of a rebound in valley fever cases above and beyond what we would have expected if the drought had not occurred. And if we look at the difference between what we saw and what we expect would have happened without the drought, we can indeed see that we are estimating an aversion of cases during the drought period, but a sharp rebound in cases following the drought period, and this rebound more than offsets the decline that occurs during the drought period. And so there are a few hypotheses for why this might be occurring. One hypothesis relates to the fact that during drought, other microbial competitors are killed off, and so when rains do return, coccidioides can grow largely uninhibited by competition. Another hypothesis relates to the fact that rodents are hosts for coccidioides, and they die off during drought, and then their bodies are serving as sources of the fungus when the rain occurs. And I'll loop back to the rodent hypothesis later on in this presentation. So this initial work helped us understand that a drought followed by wet conditions is producing conditions that are highly conducive to valley fever incidence in California. And what is concerning is that we are seeing a lot of swings in California between extreme drought conditions and extreme wet conditions. And these dry-wet swings, what's known as hydroclimate volatility, are expected to increase in California with climate change. And so the work that I had just presented kind of stopped at 2020. But of course, we are well beyond 2020. And so what has happened since? So The period of 2020 to 2022 was another period of extreme drought in California. In fact, it was capping off the driest 22-year period since 800 CE. And if you looked at the Central Valley of California during that time period, it looked much like this picture on the right from our field site, very, very dry. Then in the winter of 2022 into 2023, California experienced a series of atmospheric rivers. That winter was among the top 10 wettest seasons ever recorded. Precipitation was 150% of average. Our same field site, as you can see here, looked dramatically different at that time. And so we were worried that valley fever would be high in the following year because we had that drought period followed by increased precipitation. And just to highlight here, sorry, I skipped over it. On this graph here, you're seeing the time series of valley fever cases in California, and the lighter orange colors is the periods of drought. So you can see clearly that cases are suppressed during drought, but rebounding after. And so we suspected high number of cases. We worked with CDPH to put out some alerts. And indeed, in 2023, we saw a very large peak in valley fever. It was at the time the highest number of cases of valley fever ever recorded. And then that following winter, so 2023 into 2024, was also a pretty wet winter as well. Precipitation was 115% of average. And so this time we said, okay, let's actually get out ahead of this. And instead of just saying we think it's going to be a big year for valley fever, let's generate some actual numbers. And so we adapted our ensemble model to generate a forecasting model for 2024. This work was led by Simon Campanori and Alex Heaney. And so we took our candidate algorithms that we had before. We slightly modified our cross-validation approach using leave out one future year cross-validation rather than just leave out 1 year. And then as before, we computed a weighted average of our predictions based on the average of our candidate algorithms and how they were performing. And so this work has been published in Emerging Infectious Diseases. But what you're seeing here in blue, prior to that dotted line, is the ensemble model fit to historical data. The black dots are observed data, and the blue line is our model fit. So we were pretty happy with our model fit. And then the purple is what we're forecasting. And then the X's over top of the purple are the cases that occurred after we generated our forecast. And so what you can see from this graph here is that our forecast was actually quite accurate in predicting the number of cases of valley fever that was going to occur following that wet period after drought. We work closely with California Department of Public Health at this time in terms of sharing our forecast with them, and it did lead to several state health awareness messages, including this alert that you see here, where physicians are encouraged to suspect valley fever among people showing up with pneumonia-like symptoms in California. Okay, so that kind of sets the stage for how some of the meteorological fluctuations affect incidence in California. And I want to shift gears a little bit to talk about some more shorter term exposures, including dust and dust generating events. So because these fungi live in the soil, it is logical to think that dust and dust generating events might be co-occurring with aerosolization of these fungal spores, and that exposure to dust might be co-occurring with exposure to soil. However, there has been a lot of controversy around what the association between dust and valley fever is, in large part because it is not that easy to measure dust. So several studies had looked at the association between particulate matter 2.5 and valley fever and not finding much association. Other studies had looked specifically at dust storms and didn't find much association. And so there was a lot of confusion about how much dust really mattered. Amanda Weaver, who graduated from UC Berkeley, did her PhD work looking at estimating the exposure-response relationship between ambient dust and valley fever in California and produced kind of the first robust evidence that ambient dust is indeed associated with valley fever incidence. And what was unique about this paper is that instead of using particulate matter 2.5, she used a novel product that partitioned particulate matter into its component parts. And so she was able to specifically look at exposure to soil dust. And so the graph on the right that you're seeing here, which I'll show here. The x-axis is showing the dust concentration, and then the y-axis is showing the incidence rate ratio associated with changes in that dust concentration. And the line is showing that exposure response relationship. So what you can see here is this nonlinear but increasing association between dust and valley fever, whereby an increase of about 1 microgram per meter cubed in dust concentration is associated with a roughly 60% increase in valley fever incidence. And so once we have sort of established that ambient dust is associated with valley fever incidence, we might be curious to ask about other main sources of dust in California. So one major source of dust that might come to mind that's relevant for California is wildfires. As a fire moves through an area, it mobilizes massive amounts of soil that are carried in fire-induced updrafts and could aerosolize soil. And indeed, there is some evidence that fires are dispersing fungi. This bottom photo is a photo from some of our colleagues at Idaho who are fire ecologists, and they're flying drones through controlled burns and sampling the smoke and then testing it for fungi and bacteria. And they have found dramatically elevated concentrations of fungi and bacteria in smoke. And they've also been able to culture these microbes and find that they are not all inactivated by the heat of the fire. Long-term, fire may also have effects on dust emissions, in part because it depletes the vegetation of an area. It also can deplete the soil crust and organic matter, and so make soils more susceptible to wind erosion. It may also sterilize the soil for some of the competitors of valley fever. And so we wanted to ask, what are the immediate and delayed effects of wildfires on coccidioidomycosis incidence among people who live near those fires? And this was work that was led by Sophie Phillips, who's now at UCLA. And so to address this, we used a synthetic control approach where we generated polygons around fire burn areas in California. And then we came up with a set of controls for those, what we're calling treated areas. So the fire of interest, we included those that were at least 2,000 acres and did not overlap with another fire. And then as eligible control areas, we included areas that did not have a fire in that same time period. We then generated our synthetic control by matching the control areas to the treated regions based on the number of cases of valley fever that were observed prior to the fire in the 3 years prior, as well as other potential confounders, including population size, average climate conditions, vegetation covers, things like that. And so what you're seeing here on the right is the match of our treated unit, so the fire exposed unit in the solid line, to our synthetic control in the dotted line. So we achieved a pretty good match before the fire. And then we looked at what happened after the fire to see whether the two trends in valley fever incidence diverged in the exposed unit as well as the control units. And so what you're seeing here is that across the fires that we looked at, there was a bit of an increase in valley fever cases in years 2 and 3 following the fire, but it wasn't statistically significant. So these gray shaded regions are the confidence interval around that kind of no fire counterfactual prediction. And so what you're seeing in the treated unit is not exceeding what might be expected. Overall, fires were associated with an 8.6% increase in cases, but again, the confidence interval spanned the null. We did look at the effect of 19 fires, and as I mentioned, the pooled effect was non-significant. However, there were 3 fires that did have significantly increased valley fever case counts following the fire. And these three fires did have several things in common. First, they had higher transmission pre-fire than most of the other fires that we studied. They were also near areas with higher population as compared to the other fire studies. And finally, they were larger. So they were all over 5,000 acres, which was larger than most of the other fires that we examined. So beyond fire, another major source of dust in this study region is the drilling of oil and gas wells. So Kern County, which is that county in the southern San Joaquin Valley that I mentioned is the most endemic for valley fever, is among the top 7 counties for oil production in the country. And we do know that the drilling of new oil wells is associated with increased particulate matter and other air pollutants, and also could be increased dust emissions due to the process of leveling the ground, bringing in equipment, and then installing the well. And so we wanted to ask within Kern County, is the drilling of new oil and gas wells associated with increased risk of valley fever among people who live nearby? And so this work is now available as a preprint and was led by Lisa Cooper. So she used a case crossover study design to examine this research question. So essentially looked at when a case of valley fever occurred in California, and then looked at some hazard period prior to case onset to see whether or not oil or gas well was drilled during that period. And then she compared the exposures in this hazard period to exposure to oil and gas well production in control periods that either occurred before case onset by 1 year or after case onset by 1 year. In order to do the exposure assessment, we looked at the number of wells that were in production period within some 5-kilometer radius surrounding the case's residence. And so what she found was that the odds of valley fever among those who were living within 5 kilometers of a newly drilled oil and gas well was 11% higher than the odds of valley fever among those who did not. So here we're treating the exposure as just binary. So asking, was a well drilled in the hazard period within some buffer around the house, yes or no? You can see that the effect estimate or the odds ratio is slightly larger if we're looking at wells that were drilled closer to the house compared to wells that were drilled a bit further away from the house. If we think of the exposure as continuous, so counting up the number of wells that were drilled within some buffer around the residence, we also see a significantly positive effect. So here, for each additional well drilled within 5 kilometers of residence, we see an odds of valley fever that is 0.7% higher. And then if we look at quartiles of exposure, we see that individuals who had the highest quartile of exposure to newly drilled oil and gas wells had a 41% higher odds of valley fever than those who had no exposure to drilling of oil and gas wells. So this is all epidemiologic evidence that supports the idea that exposure to dust or dust-generating events may increase cases of valley fever. But one of the things that I really enjoyed doing in research is to combine epi-evidence with field measurements to better understand exactly how coccidioides is behaving in the environment. And so previous efforts to detect coccidioides in air have been quite limited. It is rather hard to detect coccidioides spores in the air. The most successful efforts previously have used these very large active samplers that are sampling thousands of liters of air a day and might be operating for 24 hours or more at a time. They are stationary and they are quite expensive. There has been some work to pilot these passive air samplers. So these look more just kind of like Petri dishes where dust can settle, and then the Petri dishes are tested for presence of coccidioides DNA. However, these efforts, while they are lower cost, have not yielded as much detection. And so with the Environmental Protection Agency, we were interested in testing out some new portable air filtration methods that could be used to detect coccidioides. So specifically, we worked with that Idaho group as well that I mentioned earlier that flies drones through controlled burns to understand whether we also could use these uncrewed aircraft devices to sample coccidioides. So there are some benefits of this. We could access various heights of the air column. We can send them through into hard reach areas, including controlled burns. They do collect detailed spatial information, so they are quite spatially precise. And so we mounted these low volume air samplers to these uncrewed aircraft systems that sampled air for about 6 to 15 liters per minute. And then we also had these portable ground-based samplers set up at around air breathing level. So we piloted this approach in the Carrizo Plain of California, which is a region of California shown in the red box on the left. It is a very endemic region for California, and our prior work that I will talk about in the final part of this presentation has confirmed that coccidioides is in the soil. So we flew 14 drone flights through this study region. We had a dual sampling design. So we conducted 15 flights where the drone was flown at about 10 meters altitude above the ground under just ambient conditions. And then we had 26 flights where we did this sort of paired drone design where the lower drone was flown at about 1 to 3 meters so that the wind of the drone's blades would kick up dust, and then there was a drone downwind of the first one that was capturing the dust particles that were stirred up by that lower drone. So just to kind of put some images along that second simulated high wind event, you can see the lower drone here on the photo on the left as well as in the video. And you can see some dust being kicked up, and then downwind is that second drone that is sampling from the dust that is being generated. So this work has been published in Environmental Science and Technology Letters, led by Molly Radosevich. And we were successful in identifying coccidioides immatus DNA in about 5% of our air samples. Surprisingly, we only detected coccidioides DNA in the ambient air samples, which was not what we expected. We actually expected higher detection in the flights where we were specifically kicking up dust. But our sample size was quite small. We weren't really powering this to test a hypothesis. We were more just doing it as a proof of concept. Okay, so finally, I'm going to transition to some other field research, trying to understand the distribution of coccidioides within the soils of California. So up until this point, I've really just been talking about valley fever as a disease of humans and the effects that it can have in humans. However, a human is not the only host for valley fever. And well, I will back up and say within the host, there is this second life cycle that occurs. So after a spore is inhaled, it goes into the lungs of a human. And if a human is to have asymptomatic disease, the spores are usually just kind of successfully walled off in granuloma. But for people who experience symptoms, the arthrochonidia, the spore that's in the lungs, can replicate, forming these larger endospores that can rupture and repeat this process again and again. And so a human, of course, is going to keep that spore contained in its body. However, what might happen if that host is a rodent who is living and dying in soil? So is it possible that that rodent, when it dies, could reintroduce that spore back into the soil? So this forms the basis of what's known as the small mammal endozoan hypothesis, which has really been championed in recent years by John Taylor and Bridget Barker. But the basis of the hypothesis states that rodents are asymptomatic hosts that contain spores in granulomas inside their lungs. And then when a rodent dies, potentially inside a burrow, these granulomas dissolve and release the spores. And then the carcass of that rodent serves as that primary source of nutrients for the fungus, for that spore to germinate and become mycelia again. And there is substantial evidence for this hypothesis on the basis of the fact that it is easier to find coccidioides if you are sampling rodent burrows. And there is some evidence that shows that genes of the Indigonales fungus, which is the family to which coccidioides belongs, are upregulated to degrade animal keratin as opposed to plant vegetation, which does suggest that coccidioides gets its nutrients from animal protein as opposed to plants. So we wanted to ask, what is the role of burrowing mammals in shaping the distribution of the pathogen within the environment? So as I mentioned, burrows have been associated with coccidioides, but to date, it's been really hard to understand whether the association between burrows and coccidioides is due to the fact that burrows themselves have really nice soil conditions, or due to the fact that burrows are by and large homes for rodents who live and could die inside those burrows. And so that's where the carrizo plane enters. So I mentioned the carrizo plane earlier in this presentation with respect to where we did our air sampling, but it is a national monument established in California in the Clinton administration due to its high prevalence of endangered species in California. So it is home to a great many number of endangered species, most notably this rodent that you see in the top right, or sorry, the top left, which is a kangaroo rat, called so because they hop like kangaroos and have big tails. But the kangaroo rat is really the keystone species in this area, and they are considered ecosystem engineers because they build these large mounds called precincts that really distinguish the landscape. And other rodents and burrowing animals will use these precincts as their homes as well. What you're seeing in the middle is a view of the carrizo plane from space. In 2007, the carrizo plane ecology project was started, in which researchers who were interested in studying the effects of cattle grazing on rodents developed these experimental plots. And in the center of the plot, they developed these rodent exclusion zones. So you can kind of see in the middle of this plot a smaller square. That is a rodent exclusion zone. Chicken wire fence was dug 2 meters below the soil and then extended above the soil. And any rodent that was in that exclosure was trapped and relocated. And so what that gives us is burrows that have been abandoned of rodents since 2007, allowing us to do this nice natural experiment where we can compare coccidioides in burrows with rodents to coccidioides in burrows without rodents. So since 2020, we have been sampling seasonally in the carrizo plane across this factorial design that crosses burrows and exclosures with surface soil and exclosures. So we have collected about 500 samples per each sampling session and done about 2 sessions per year. Within each plot, and there are 20 plots in total, we collect soil from 5 clusters, including 2 burrow systems outside the exclosure and 1 burrow system inside the exclosure. And then we also collect soil from the surface nearby these burrows, both inside and outside the exclosure. Along with the soil, we collect information on soil moisture, soil temperature, and vegetation level. And then there are other groups from Cal Poly who do rodent abundance surveys. So in order to understand the effect of rodents on coccidioides in soil, we applied this causal mediation analysis with G computation. And really what we're interested in here is first, the overall effect of rodents on coccidioides. So the effect of rodents due to the fact that they, 1, create burrows, but 2, could be contributing to coccidioides presence in soil either through the they're dying and reintroducing spores back to the soil, or just the shedding of keratin, which is providing nutrients to the soil. We're also interested in the indirect effect of rodents on coccidioides, which here we're defining as the effect of rodents on coccidioides that is operating via burrows. And then the direct effect, which we define as the effect of rodents that is not acting via burrows. And so I'll say that this could be due to that endozoan hypothesis, whereby an infected rodent dies and reintroduces the spores, but it could also be to other mechanisms like the fact that rodents provide more nitrogen to the soil just through their through defecating or by shedding of hair and skin and things like that. So I'll skip this slide. This is just our model for our mediation analysis approach. So on to our results. So far, we have analyzed data across 2 years of sampling, which represents about 2,000 samples, and we detected coccidioides DNA in just over 16% of samples. We found the highest percentage of samples with coccidioides among samples that had that were burrows and had rodents in them, about 30%. About 19% of burrow samples from the rodent exclosures, where rodents are absent, were positive for coccidioides, and then much fewer surface soil samples were positive. About 1% of surface soil samples out inside the exclosures, and then about 5% of the surface samples where the rodents were present were positive. When we did just a generalized linear mixed model where we regressed odds or a binary indicator for coccidioides presence against variables for soil moisture, burrows, and presence of rodents, we see that both burrows and rodents are significantly associated with the presence of coccidioides in the soil. Specifically, rodents are associated with about a 2.6 times higher odds of detecting coccidioides immitis compared to no rodents. But burrows are really more strongly associated. So the odds of coccidioides in burrow soils was about 19 times higher than the odds of coccidioides in surface soils. And then soil moisture was also associated inversely with coccidioides. When we put this into our mediation analysis, we saw that overall the odds of coccidioides is 32 times higher where rodents are present compared to where rodents are absent, and that about 3/4 of the effect of rodents on coccidioides presence is mediated by burrows, meaning that about 1/4 is not mediated by burrows, so could be due to direct effect of rodents themselves. When we added soil moisture and soil conditions to this model, we see that about 8% of the effect of burrows on coccidioides could be explained by lower soil moisture in the burrows. So we are not really getting at the soil conditions that make burrows more conducive for coccidioides here, but we do plan to work closely with other collaborators from Texas to understand more about soil nutrient profiles and how they might differ between burrows and surface soils. And explain why burrows might be such suitable microhabitats for the fungus. So I think it's worth mentioning why it matters knowing if rodents are involved, beyond just the simple fact of it's interesting. So first, we've talked about how people who work outdoors might be at increased risk of valley fever in California. California does consider valley fever an occupational disease, which is different than Arizona, who does not. But there are only regulations to protect construction workers who dig in the soil, even though there have been outbreaks among other occupational groups that dig near rodent burrows. And so if we knew that rodent burrows were specifically occupied rodent burrows were high risk, we could introduce more regulations for people who are digging near rodent burrows. It's also helpful to contextualize climate influences. Earlier, we talked about how in California, valley fever is suppressed during drought, but then rebounds sharply following drought. And these dynamics are actually quite similar to hantavirus, which of course is a rodent associated disease as well, where we see outbreaks of hantavirus following drought periods when rains returns. And a lot of that is associated with sharp drop-offs of rodent populations during droughts, but then a sharp rebound in rodent populations populations following droughts when rains do return. And then finally, it helps us to understand geographic expansion. So in the graph on the top, what you're seeing here are predictions from Morgan Goris about how valley fever is expected to expand across much of the Western US by the end of the century. But of course, this graph is just showing what habitat is suitable for the fungus. It can't actually tell us how the fungus might actually move to those new areas. But we do know from other research that both wind and small mammals are capable of dispersing fungi. And so we are quite interested to know whether coccidioides might be spreading more effectively in the wind or within the bodies of these small mammals. So just to summarize the work that I've presented here today, we first found that hydroclimate swings appear to amplify incidence in California. So within a year, we see that wet winters followed by hot, dry summers amplify incidence, which makes sense if we think about how wet winters are fueling the growth of the fungus in the soil, and then dry summers are allowing the soil to desiccate, the spores to become disarticulated, and then the soil to become eroded. However, if we look across more than just a single year, we see that drought followed by wet winters is associated with incredibly high incidence in California. And we can use these antecedent meteorological conditions to generate fairly reliable disease forecasts that can be useful for public health to alert physicians to increase suspicion for valley fever. We have seen that ambient dust is associated with incidence. Wildfires, a major source of dust, are not by and large associated with incidence, but we do see some evidence to suggest that perhaps larger fires in areas where the disease is actively circulating may amplify incidence as long as 3 years after the fires. We do see evidence that drilling of new oil and gas wells is associated with higher incidence within Kern County. And we postulate that new methods of airborne spore detection might help us better understand how coccidioides are aerosolizing and transporting. Finally, we see that coccidioides is 32 times more likely to be detected in the soil when rodents are present compared to absent. Most of this effect is due to burrows, which suggests that the burrows are suitable microhabitats. However, we do see that rodents themselves are also amplifying presence. We have partnership with Texas A&M to hopefully reveal the role of other soil conditions in this relationship, like nutrients. And one key limitation to note in all of these field studies is that the methods do not actually tell us if the fungus is viable. It just tells us if the DNA is present. So we aren't able to say whether the fungus we're detecting is actually living fungus or whether it's just been hanging out there for a long time but is not actually active. So that's all I have for valley fever. I do want to put out a post for this community that if you are looking for a postdoc, we are launching some work here at Michigan about H5N1 transmission among peri-domestic mammals. So if that's something that you're interested in, please contact me at this email address below, and I would love to chat more about that. And so I'll just, again, put up my acknowledgement slides, and thank you all for listening, and I'm pleased to take any questions.
Thank you, Jennifer. We'll just open it up for questions. Feel free to either put it in the chat or unmute yourself, or raise your hand, and then we will start figuring out who's first, second, and third.
Hi, Jennifer. Erin Mordecai. Great talk. That was really interesting. I remember seeing you present this work at Stanford a few years ago, and it's just amazing to see how much has happened since then. So I was really impressed by the performance of the forecasting model that you developed, and I was curious whether you dug further into which factors that were included in the model were contributing most to that forecast accuracy, whether that can tell you anything about what the key environmental drivers are.
Yes, that's a great question. So when we built this forecast model, we purposefully did try to keep it simple because we rationalized that a forecast model, in order for it to be useful, it shouldn't involve collection and summarization of a lot of environmental data. And so our main terms that were in the model was just temperature, lagged by up to 36 months, and then precipitation lagged by up to 36 months. And so the most important variables that were in the model was the precipitation in that kind of 5 to 10 month period before, which is actually really nice for a forecasting model because it lets us get pretty accurate predictions like 4 to 10 months in advance. And then in the random forest model, some of that interaction between precipitation, like in the, 24 months past, and then in that 5 to 10 months was important. So I think in California, at least precipitation is really important. We are doing some work in Arizona as well. And I will note that Arizona is a little bit trickier because it has 2 rainy seasons as opposed to 1. And so forecasting in Arizona is just a little bit more difficult, but even there, precipitation seems to be one of the most important drivers and predictors of incidence.
Really cool. Thank you.
Looks like I have a chat question.
Yes, okay, so the question says, do you have an idea which small mammals were most common in the trapping data aside from the K-rats? Imagine mammal community structure could also impact the incidence. Yeah, so in this ecosystem, the giant kangaroo rats really dominate as the ecosystem engineers. The other species that we see commonly are the San Joaquin antelope squirrels, which are these much smaller bodied species, and they do occupy those precincts that are built by the GKR. During drought periods, you do see some rise of other small-bodied mammals, but by and large, about 90% of the species in this area are either the giant kangaroo rats or the San Joaquin antelope squirrels. And the giant kangaroo rats, from what I've heard from the Cal Poly folks, tend to not move as much between precincts. They generally have a pretty smaller home range, but the San Joaquin antelope squirrels might move quite further across the landscape.
I had 2 questions for you, Jennifer. One was, do you have any sense for what the fraction of cases are that are reported versus unreported?
Yes, that's a great question. So valley fever it's funny, it's considered a rare disease, but if you go to parts of California, like the Central Valley, and mention valley fever, almost everyone has a story to tell about how they've gotten valley fever or their friend has it or dogs have had it. So it's really not rare in certain parts of California. We do think that about 60% of cases are asymptomatic. And then of the remaining 40%, 1 to 5% might become chronic, and then 1% might disseminate throughout the body, causing very severe infection. So a lot of these mild cases are probably missed. We also know that there's a lot of misdiagnosis of valley fever because it presents very similar to pneumonia. And so it is estimated that roughly about 1/3 of pneumonia cases are actually valley fever cases in these endemic regions that are just not identified as such. But recently there was a study done by folks from CDC's mycotic branch that predicted that we're capturing about 1 in 10 to 1 in 18 cases of valley fever nationally. Within highly endemic areas, the capture rate is a little bit better, but still about like 1 in 6 symptomatic cases are estimated to be captured.
My other question was, for the wildfire study, what's your sense for like how far those plumes of smoke go, and how do you handle that with like getting like not having contamination of your controls and your treatments?
Yes. So in our epi study with the wildfires, we were really just looking at that burn area. So our treated region was just whether or not it was part of the burn area. So we weren't, I suppose, looking so much at smoke exposure, but more just at like very acute exposure to being near that region and then being near that kind of like charred area afterwards. There have been other studies that have looked more at smoke plumes as the exposure and did find associations between smoke plumes and and valley fever incidents in California. So that's just one other source of epi evidence. The folks from Idaho who do some of the field sampling of smoke, I think they're pretty close to the controlled burn. Um, and they do controls as well because it's part of the controlled burn. So they collect samples before the burn starts and then collect samples during the time of the burn, if that answers your question.
We have a
And
question in the chat from Sean, who asks, it doesn't seem that there's many cases in southeastern California in the Mojave and Sonoran deserts. Would you say this is due to aridness and low precipitation in the region?
Yes, I think that's a great question. So that would be my hypothesis. So I didn't show so much these nonlinear relationships with precipitation um, and valley fever, but it does seem like there is kind of a sweet spot where more rain is conducive to valley fever up until a point, and then after some point it maybe becomes a bit too wet. And so I do think that the incidence is lower in these areas, and I do suspect that it's because there's just not enough moisture for the fungus to grow. I see, yeah, Kendra suggesting lower population, which is definitely true, but incidence is also quite a bit lower in this the desert regions. I will say though that the species of Coccidioides in Arizona is different than the one in California. It's Coccidioides posidacei is compared to Immitis, and it seems to be a bit more thermotolerant and perhaps thrive a bit better in drier conditions. And so in Arizona, the Sonoran Desert is a very high hotspot for valley fever. And it may be that the fungus is tolerating some of the drier conditions a little bit better than the California species.
Tanner, did you have a question?
Yeah, great talk. Thank you so much. I'm curious if there was any justification for the 5 kilometer radius around your oil and gas development, and if you considered going out a little bit further on that, or if it's just more and more like the larger the distance, the harder it is.
Yeah, that's a great question. So part of that was motivated by some prior work from David Gonzalez, who's at UC Berkeley, who has looked at the increases in particulate matter during either the drilling of new oil and gas or the increase in production in oil and gas, and found increased particulate matter roughly up until that 5 kilometer distance. But I do think, because we are seeing effects still at that 5 kilometer distance, that it, could be interesting to look further out from that.
Thank you.
Maybe with that, we'll like pause or halt the video